and then go back to the share. Okay, so the first slide I had for chapter 9 was all the way up to 18. So the first thing we talked about in chapter 9 was the replication of DNA. So there are different DNA polymerases. You don't have to know those, but you should know just the basic structure of DNA. It's a ribose sugar, phosphate, and 4 nucleic acids. It's a double-stranded molecule, and you should know that A binds to T, G binds to C in DNA. And excuse me, for synthesis of the molecule, it has to open up, and you get a daughter strand and a parental strand. So the daughter strand would be the new stranded DNA. The molecule has to unwind, separate, can only add nucleotides to an existing chain, cannot begin synthesizing a chain of nucleotides, and can only add them in the 5 to 3' direction, which was determined by the number of carbons on the ribose sugar. So that was 18. And the site of replication of DNA is called a replication fork. That's where the helix is unwound and replication takes place. So if you remember, bacteria have just a single circular DNA molecule. We have linear DNA molecules. So in the bacteria, that circular DNA molecule will have to open up and have 2 replication forks. You need a primer to get started, which is RNA, and then the RNA gets removed. Mark this. So you have a leading strand and a lagging strand. The leading strand, because it can be synthesized continuously, but in the lagging strand, the molecule has to wait to open up. It is synthesized in fragments called Okazaki fragments that later get joined together. And this is a picture of that replication fork. So this is the 5 to 3' direction. The red represents the new molecule. This represents the enzyme, the polymerase enzyme that makes the strand. So as this opens up, it can just go in a continuous fashion. But since it has to be now, remember, it's an anti-parallel structure. So the other DNA, the 5 to 3' direction, would be going this way. So as it opens up, a fragment gets synthesized. So here's one. They're starting to get joined down here. A ligase enzyme joins the fragments together. There will be a new one here. You have some proteins to help stabilize it, because DNA is very stable as a double helix, single strand not as stable. You have a topoisomerase, a helicase that cuts the bonds. That's why they have the little scissor symbol there. Topoisomerase, think of a top, it's unwinding it, because the double helix, it looks like a spiral staircase. Then I had slide number 7. We talked about the genetic code. It's the central dogma of genetics. Transcription is when DNA is used to make RNA, and translation is when the RNA is used to make the proteins. There's a wide variety of RNAs that are used to regulate gene function. They can even be involved in human disease. The DNA that codes for these crucial RNA was once called junk DNA because people didn't seem to think it had any function. Traditionally, the RNAs that we know about are the transfer RNA, the ribose RNA, and the transfer and the message RNA. So the genetic code is found on the DNA. The codons are found on the message RNA. They're transcribed from the DNA. Those codons are what you use to read the genetic code. There's a corresponding anticodon on the transfer RNA when it matches, in other words, complementary binding to the message RNA codon. It carries the amino acid. The ribosomal RNAs combine with ribosomal proteins to make up the ribosomes. So the gene-protein connection, the connection between a DNA and an organism's traits. The protein's primary structure determines its characteristics, shape, and function. So when protein's first made, it's just a long strand that's called the primary structure, can then have a secondary, a tertiary structure, and then it can bind with other proteins to make complex quaternary structures, which could be true of enzymes and structural proteins and signaling molecules. But proteins ultimately determine an organism's phenotype. That's the genes that are actually seen or expressed. Another area of science that has become a big area of interest is, instead of looking just at the genomics, we look at what we call the proteomics, and that's the complete set of actual proteins that are expressed. Not all of your genes are expressed. Some of them might be recessive, and they might even skip a generation. But your actual phenotype is what all your genes are that are actually being expressed. DNA is a blueprint that tells a cell which kind of proteins to make and how to make them. Simplified view of the DNA-protein relationship. We start with the DNA. So the genetic code is in triplets. And by complementary base pairing, we get the message RNA has to match, when this opens up, match those that genetic code. Each triplet of nucleotides equals an amino acid. And it also determines the order of the amino acids. So if this codon represents this circle, that's going to be the first amino acid, second, and so on. Message RNA is very important. It determines the sequence of the amino acids. It transports the DNA code to the ribosome, and it is translated. It is the only RNA that is translated, the only RNA that has the codons that represent the genetic code. The transfer RNAs, I said, have the anticodon. They are not translated, but they bring the amino acids to the site. Protein synthesis. The ribosomal RNAs are made up of some structural RNAs along with some proteins, the only RNAs that bind to proteins. Now, there were some other classes of RNAs. We briefly talked about these other RNAs do not play a role in protein synthesis. There's what we call the microRNAs. They're regulatory. They can regulate gene expression. There's also these small, what we call small interfering RNAs. These are a big area of research right now, and possibly fixing some genetic defects. There's the RNA primer that's part of DNA replication, obviously not part of protein synthesis. And then there's something called ribozymes and spliceosomes. These are our actual RNA enzymes, and they're part of the splicing enzymes. We mentioned that in eukaryotes, the genetic code has interrupted sequences called introns, and they are removed by these special RNA enzymes. This is only something seen in eukaryotes and not in prokaryotes. Tools in the cell's assembly line, differences in RNA structure. Single-stranded molecule that exists in a helical form can assume secondary and tertiary levels of complexity, but generally, even with those more complex structures, like if you remember the transfer RNA had this clover leaf structures, those are, it's still a single molecule, just assumes some other levels of complexity. Also remember in RNA, uracil replaces thymidine. Thymidine only found in DNA, uracil is only found in RNA. The other major difference with RNA, the sugar is ribose, where in DNA it's deoxyribose. So DNA is deoxynucleotide, and RNA is ribose nucleotide. So the messenger RNA obviously carries the message. It's synthesized in a process similar to the leading strand synthesis in DNA replication. It has the codons for the amino acids. Transcription initiation. So the molecule, the DNA molecule, has to open up. Now, this is not replication. This is the beginning of transcription. You have an RNA polymerase, the enzyme that makes the RNA. You have an initial start codon. TAC is the start code in the DNA. The master genetic code, the message in the message RNA, the central principle of translations. The message nucleotides are read in codons in groups of 3. The codon dictates which amino acids are added to the growing chain. Except for a very few cases, the code is universal for bacteria, archaea bacteria that we only talked a little bit about, eukaryotes, and viruses. And now with this new technology in making vaccines using message RNA, this is brand new technology. It's actually been worked on for a long time and always thought to be theoretically possible. And we now have 2 vaccines, and it uses the virus message. And when it goes inside your cell makes the viral proteins. And when the proteins go out of the cell, your immune system reacts to them as foreign. So we mentioned the genetic code. I mean, this is it. This is your entire genetic code. It's pretty universal. So if this is true for you, it's true for cats, dogs, trees, bacteria, fungi. We mentioned that there was redundancy. So remember, this is being read from the RNA, so there's no uracil in DNA. So there's redundancy in the genetic code. What's most important is the first 2 nucleotides. You see that in the case of the phenylalanine, the first 2 nucleotides are the same. The third one is different. You can see that there's a lot of codons for leucine. So if there's a mistake, let me see, make sure I met these other people. So if there's a mistake in this third position, we still get the right amino acid. The AUG is the start codon. It codes for F-methionine. Not every protein begins with this. It can actually be removed, but you have to have the start codon. You see we have 3 stop codons. Stop codons do not code for an amino acid. They mean what they say, they are stop codon. Oops. So we mentioned there's redundancy. Certain amino acids are represented by multiple codons. It allows for the insertion of correct amino acids even when mistakes occur. We refer to that third position in the codon as the wobble. Only the first 2 nucleotides are actually required to code the correct amino acid. The third one does not change its sense. In other words, it still makes sense that what amino acid it codes for. So this permits some variations due to mutation without actually altering the message. Interpreting the genetic code. So again, starting with the DNA, TAC would become on a message AUG because T is complementary to A to U. There's no T in the RNA, C to G. So AUG is your start codon. And so then the transfer RNA is going to have an anti-codon. They'll be complementary to that. So A binds to U, U to A, G to C. So the anticodon's UAC. So AUG is that first amino acid. CUG is leucine. Threonine is ACU, and ACG is also. You see both of these begin with AC. The third codon is different, but it gives you the same amino acid. Translation, the second stage of gene expression. All the elements needed to synthesize a protein. You have to have the message RNA, transfer RNA, the amino acids, and the ribosomes. Translation is in 3 stages: the initiation, the elongation of the protein, and then eventually the termination in a mature protein. For those of you that came in late, there's the study guide in module 3. I have a list of what I thought were the most important slides as they relate to the test. So that's what I'm reviewing. Okay, so in eukaryotes, the introns are removed at the site of the junction between an exon and intron. This special RNA enzyme that we mentioned gets involved. This becomes a spliceosome. These introns are removed. The exons are joined together. The complete message RNA can then proceed to the cytoplasm for translation after the removal of the introns. This does not happen in bacteria. They do not have introns in their genetic code. So operons, we talked a little bit about regulation at the level of DNA. One of the first ones found was the operon, which is found in bacteria. An operon can coordinate a set of genes as a single unit. It can either be turned on, which means inducible, means it's turned on, or the gene can be repressed. Categories determine how transcription is affected by the environment surrounding the cell. So catabolic operons, encoding enzymes that act in catabolism, are inducible. An operon is turned on by the presence of the substrate that needs to be metabolized, for which the structural genes encode. Enzymes are needed to metabolize a nutrient, are only produced when the nutrient's present in the environment. Actually, it didn't include this end slide, but let's go to 67. Now, bacteria can do something unique. It's called phase variation. So bacteria can turn off or turn on a whole series of genes, and they can actually change their phenotype. This phase variation is heritable. It can be passed to other generations of bacteria. It involves turning on genes mediated by regulatory proteins, as described with operons. This gives bacteria a selective advantage in survival by completely changing their phenotype, depending on what's going on in the environment. DNA recombination events. So this kind of transitions us to the DNA technology. Recombination is an event in which one bacteria donates DNA to another. So anytime you introduce outside genetic information, it's a recombination event. The end result's a strain both different from the donor and the recipient's strain. Plasmids, if you remember, these are small extra chromosomal pieces of DNA that bacteria have. They can carry virulence factors. They can be readily exchanged between bacteria, and sometimes they can integrate into the genome. Recombinant, any organism that contains and expresses genes that originated from another organism is a recombinant. So in bacteria, so our gene transfer is vertical. It goes from one generation to the next. Bacteria can do it horizontally, and they can acquire exchange genetic information between them. So it does not, the new genes do not come directly from a parent organism. These plasmids are small circular pieces of DNA. They replicate independently of the bacterial chromosome. We mentioned they allow transfer of DNA between cells. They are found in most bacteria. They are found in some fungi. The plasmids carry few genes, maybe a few dozen genes at the most. They are not necessary for the survival of the bacteria, but they can carry useful traits, such as antibiotic resistance, resistance to heavy metals. We talked about these three processes of horizontal gene transfer in bacteria. These have been utilized by researchers in DNA technology. The one major category was conjugation, requires direct contact. a drug, a donor cell with the genes for a fertility called I'm sorry, I mangled that, fertility plasmid, has genes from the donor cell to make something called a sex pelus. And this establishes a bridge between two cells and allows for transfer of DNA. So we already mentioned drug resistance. It can also contain toxins, adherence molecules, degradation of things that are toxic to the bacteria, and also to take up iron. Transformation was an early process that was identified when a researcher was looking at some bacteria that were virulent, caused pneumonia, and some that weren't. And it turned out the virulent ones had a gene to make a capsule, which helps protect the bacteria from phagocytosis. So when a bacterial cell is broken open, if it's a competent, it releases free DNA, and if the other cell is a competent recipient cell, can actually take up DNA from the environment. You get recombination, and you get the genes to make the polysaccharide capsule. Another way genes can be transferred is called transduction. This involves a virus that infects bacteria called a bacteriophage. And sorry, I got somebody to admit. The donor cell can be lysed by the bacteria, by the bacteriophage. It can pick up some of the bacterial DNA and deliver it to another bacterial cell. There were two types. Of transductions events, specialized and generalized. So generalized, just random pieces of bacterial DNA is transferred, and specialized, only where the virus inserts its DNA into the bacteria. So where are we? So I'm going to go to 85. So to review those, specialized transduction, you get integration of the virus DNA into the bacterial chromosome. It's called a prophage. We have something similar that can happen in us when a virus DNA gets into our cells, like the chicken pox or the AIDS virus. In the bacteria, it's a prophage, because that's what the viruses are called, bacteriophages. In us, it would be called a provirus. So when this, in specialized, when this DNA comes back out, it'll contain on either side some bacterial DNA, new viruses get made. They eventually leave the cell and affect other cells, and they can now carry some bacterial DNA from this original cell up here into a new bacterial cell. Mutations, we talked a little bit about, or changes in the genetic code. And there's, mutations can be either positive or negative. They can give an organism a selective advantage versus other organisms. They can also lead to defects that make the organism less able to survive. They can be negative. Some major categories of mutations are frame shift. That's the reading frame of the genetic code. You get a nonsense mutation. A silent mutation is when there's really no change. But remember that third place on the codon can change. That can be a silent mutation. You still get the same amino acid, but the codon's a little different. And lastly, you can have missense mutations. So any change in the nucleotide sequence is a mutation. It is the driving force of evolution. In microorganisms, mutations become evident in altered gene expression, such as altered pigment production or development of resistance to a drug. We refer, and this isn't just bacteria, we refer to the original organism as the wild type. So, for example, the COVID-19, when it first was discovered in its sequence, when it was sequenced back in January of last year, that would have been the wild type. We now have all these variants because of mutations. So eventually, this wild type could be replaced by a mutant strain. In other words, there's been a genetic change. The mutant strain actually might have a selective advantage over the wild type, and the mutant strain then becomes the dominant organism. Mutations can happen spontaneously during errors that occur in replication. In various mutagens, these are induced something like chemical or physical reaction from the outside that affects the DNA, can cause the mutation. Selective, do I have this on here? I actually don't have anything about the different agents. There's chemical agents, there's radiation, ionizing, which is gamma and X-rays, and non-ionizing, which is UV light. These can all affect the structure of DNA. Now, point mutation affects only a single base on a gene. It involves either the addition of a nucleotide or the deletion or the substitution for a different base. A lethal mutation, some mutations are so bad they can lead to dysfunction and death of an organism. A neutral mutation produces neither adverse problems but it's not necessarily helpful. So in other words, it's neutral. A missense mutation, it's a change in the genetic code that leads to placement of a different amino acid. So that's a bigger change, an actual change in the amino acid. A nonsense mutation, the normal is when a normal codon becomes a stop codon. So the stop codons, remember, do not code for an amino acid. It would also result in the truncation of a protein because it's a stop codon. We talked a little bit about how to read the genetic code. This is, actually, I don't have this on the list of slides, so I'm going to skip over that. Repair of mutations. Your body does, and organisms do have ability to repair to a certain extent. So one is photoactivation. So UV light can cause damage, and an enzyme complex can come in and cut out the mistake and replace it. Another enzyme comes in and replaces it. Excision repair, mutations are excised by a series of enzymes that remove the incorrect bases and add correct ones. Positive and negative effects of mutations. To summarize, many mutations are not repaired. Effects of mutations depend on the nature of the mutation and the strategies that have evolved that are available to an organism. Mutations are permanent, they're heritable, they're passed on the offspring. For example, new viruses become organisms and new viruses, they become a long-term part of the gene pool of these organisms. A small number of mutations contribute to success and eventually can lead to the evolution of new species, as long as they adapt, that they survive the new mutation, and that it's an advantage to them versus the organisms that don't have the mutation and are able to reproduce and pass on the new genes. Okay, so that was quick review of chapter 9. So let's go on to chapter 10. And we're going to chapter 10, start with slide 4. So this is our DNA technology. And I said some of the things that in the last section, horizontal gene transfer in bacteria, some of those things that happen have been widely used and manipulated in DNA technology to introduce new genes. So those three are transformation, transduction, and conjugation. So what is the difference between basic science and applied science? So basic science is just a simplified definition, just knowledge for knowledge's sake. But applied science is something that we can apply that's a benefit. So early on, to get to the point where we have these RNA vaccines, we had to know about the genetic code. We had to know about transcription and how translation occurs. We had to know about how viruses work and to develop the technology to take advantage of that. So we basically take advantage of the genetic code of the virus and the process of translation of proteins to make these successful vaccines. So something used in DNA technology, these enzymes that bacteria make called endonucleases that can be used to cut the DNA. A common process to use something called gel electrophoresis to analyze the DNA. A common technique is something called polymerase chain reaction. It can be used to take a small piece of DNA and to make many, many copies of it. And we'll talk a little bit about the creation of recombinant DNA and its role in gene cloning. Intrinsic properties of DNA. Obviously, the DNA is the raw material. We mentioned the helicase enzyme used to unwound the DNA. That can be used in the lab to our advantage. We know that DNA will separate when exposed to temperatures just below boiling. We know that complementary nucleotides will hydrogen bond and regain the double helix. So you can open the DNA, you can cut it with enzymes. You can then allow it to re-anneal, which it kind of does for you because of the complementary base pairing. Heat it up to separate, cool it back down to get it to form a double helix again. The enzymes made by bacteria, the restriction into nucleases, they will cut DNA at selected positions. They recognize foreign DNA. They're capable of breaking those phosphorodiester bonds. Remember, the backbone of the DNA is the sugar phosphate. And between the adjacent nucleotides on both strands of DNA, bacteria and archaebacteria use these enzymes to protect themselves from bacteriophages or even foreign plasmids. Each endonuclease enzyme recognizes a sequence of about 4 to 10 base pairs. The enzyme recognizes a site that has a palindrome on both sides. So in other words, at the very end of the sequence, it gets cut on both sides of the DNA. Is a sequence that's the same but reads in an opposite direction on one strand end of it versus the other end. When the DNA is cut, it leaves sticky ends, 4 to 5 base pairs on each strand. Base pairs can be complementary on the DNA fragments or plasmids. So there's an enzyme to then join the new DNA to the old and not to the old, to the other DNA. And the main applications, the final splicing of genes in DNA technology, either into plasmids or directly into chromosomes. Of an important enzyme from the retroviruses, HIV is a retrovirus, is they have this unique enzyme we call reverse transcriptase. They can take their genetic materials, RNA, they can use this enzyme to make complementary DNA. So they're able to convert RNA to DNA. So in a laboratory, when you make the messenger RNA, if you're starting with a eukaryote, if you remember, the introns are removed, you get the messenger RNA, goes out in the cytoplasm. And then we can, in a laboratory, we can isolate that messenger RNA. We can add the reverse transcriptase enzyme, thereby making a complementary DNA called a cDNA. We call it a cDNA library because every cell is going to be expressing X number of messenger RNAs. Not every gene gets expressed. And so you have a library of cDNAs that represent all the messenger RNAs that a cell was making. And it's also free of the introns. So gel electrophoresis, DNA, when they're cut, makes characteristic DNA fragments. They can be placed in a soft gel. An electrical current can be applied to the gel because the DNA has those phosphates. It has a negative charge and it'll move in the gel toward a positive pole. The rate of movement through DNA is based on the size. So the bigger pieces will be at the top of the gel, the smaller pieces will move faster and further in the gel. And this pattern of fragments of DNA are very characteristic. They can be used to identify individuals. It's used in forensic science. So we call that genetic fingerprinting. You see an example here. So this is a predetermined markers. You buy these from laboratories, so you can then calculate the size of these other fragments. Two different nucleotides can hybridize by uniting at their complementary regions. So single-stranded DNA can hybridize either with a single-stranded DNA molecule or to RNA. RNA can hybridize with other RNA. So we can use this knowledge to make probes to look for the actual genes. So a probe is a short sequence of DNA that will base pair with a stretch of DNA within a complementary sequence if there's one in the sample. These karyomolecules can have fluorescent dyes, so you can actually visualize the hybridization. Enzyme-linked probes can be detected when non-pigmented substrates are converted to colored substrate, colored molecules by the action of an enzyme. The use of gene probes, we can use it to diagnose the cause of an infection in a patient's specimen, identify a culture of an unknown bacterium. So if you know a characteristic gene for a bacteria, you can put a genetic probe in with a fluorescent dye and actually detect it. You'll be able to see it on a gel. So a nucleic acid hybridization test. So you start with a plate with unknown bacteria, put a filter membrane on it, transfer the colonies to the membrane, and then you treat it with a chemical to separate DNA into single strands. You then add your probe with the luminescence, and the probe will hybridize with the desired gene. If the bacteria has the desired gene that matches the probe, you then develop this, and you can then, because it's fluorescent, it can be visualized under UV light, and you can identify the colonies that have the gene. Polymerase chain reactions. So actually, this has a lot of applications. One now for detecting COVID, you can take pooled samples of individuals, and you, because the virus has messenger RNA for its genetic material, you can take advantage of that and do a polymerase chain reaction. So PCR uses the same events of DNA replication. If you start at the beginning, if you're starting with the DNA instead of the message, you open the double helix, you expose the strands, you add primers, you add a DNA polymerase, and you can synthesize it. Specialized ingredients used in PCR, you have to have some primers. These are synthetic oligonucleotides. They're usually 15 to 30 bases that will indicate where amplification of a gene will begin. You have to have some DNA polymerase enzyme. And we have now isolated from bacteria, thermophilic bacteria that like a warm environment. They have enzymes that'll survive those high temperatures. So we can use that in the process of PCR. Okay, so let me skip over to 34. Using recombinant DNA for gene cloning, take a genetic donor, isolate a gene, put it into a plasmid. Using recombinant DNA for genetic cloning, so we can use those special enzymes. The endonucleases creates these staggered cuts that allow us to put a gene into, to cut out a piece of donor DNA and then put it into a, splice it into a segment of DNA, into a plasmid. We then can put the plasmid into the bacteria using a process similar to transformation. You have to have, in the laboratory, you can either do this with a chemical to make the bacteria take up the plasmid, or actually give them a mild electrical shock that'll make the cell permeable. So anyway, they pick up this plasmid. It has a new gene in it. You then grow up the bacteria and they will make lots of whatever this product is, a vitamin, an antibiotic. I sort of jumped ahead of myself, but the bacteria with the donor gene, it will get transcribed, get translated. It'll make the protein, and you grow up the bacteria, and then you can separate the bacteria and purify the protein product that you wanted. Products of recombinant DNA technology. Well, any organism has new genes are recombinant. We can use these now to make protein products or nucleotide sequences, medications that can't be manufactured any other way. It's used in the large scale manufacture hormones such as insulin, human growth hormone, enzymes. Transgenic organisms, these are genetically modified organisms. They have recombinant genes that have been recombinant organisms produced by the introduction of foreign genes. These are then referred to as genetically modified organisms, or GMOs. Transgenic animals, we can engineer their embryos. Several hundred strains of transgenic animals have been introduced by research and industry. Unlike bacteria and yeast, animals can express human genes in their organs and organ systems that are very similar to those of humans. Animal models have been designed to study human genetic diseases. These natural systems are used to test new genetic therapies. Sheep and goat have been engineered to become factories. They manufacture human proteins, excrete them in the milk or semen. This is known as farming, as in pharmaceutical. Play on the word farm in pharmaceutical. For certain diseases, the disease phenotype is due to lack of a single specific protein. Type 1 diabetes is caused by a lack of insulin. Insulin was first for people with type 1 diabetes. It was first provided from the pancreas of pigs and cows. Recombinant human insulin was first genetically engineered drug that was approved for use in humans. Gene therapy aims to repair or correct a faulty gene in humans suffering from a fatal or debilitating disease. The benefit would be the permanent cure of a physical dysfunction by repairing the genetic defect. So the basic protocol for this in an idealized system where a normal gene is isolated from a healthy subject, you can then use DNA technology to clone that gene and insert it into a retrovirus vector. It's also itself been genetically modified not to cause disease. A bone marrow sample is taken from the person who has the defect, and it gets transfected by this virus that has the repair gene. And then the individual is infused, and they are observed to see if they have expression of a normal gene. Experimentation, clinical trials of gene testing. We've been doing this with cancer, single gene defects, and infections. Most gene deliveries are carried out by virus vectors. This isn't recombinant DNA technology, but we're able to now create some vaccines by using a virus in one that's been changed so it's not infectious, and getting it to express just some of the proteins of what you want to vaccinate. So they use a modified adenovirus. I think the Johnson & Johnson vaccine uses that technique instead of the messenger RNA. Though in this example, I'm trying to repair gene defects or target cancer, early trials were hampered by safety issues in which the virus uses delivery vehicles themselves cause malignancies. Germline gene therapy, genes are inserted into an egg, sperm, or early embryo. And the new gene will then be presented in all the cells of the individual, and that therapeutic gene would be heritable thereafter. Diseases resulting from the inappropriate expression of a protein, Alzheimer's, virus diseases, many cancers. Sometimes this occurs with a malfunctioning microRNAs. These are small regulatory RNAs, and they are targets of gene therapy. Okay, so that's chapter 9 and 10. We're going to lastly review our section on viruses. If you came in late, I set up a what's called a YouTube channel, and I have the last 3 lectures on it, but I have to figure out how to get that to you. I tried just going on Google and Googling myself. There's actually a pretty famous basketball coach, I think here in Baltimore, named Chester Frazier. So I have to maybe figure out how I can just put the link into an announcement, and you can then view the lectures that way. All right, so our last section is, so once we do chapter 6, if you have any other questions, I will entertain them. I'm also recording this, and I'll add this review to our section. Okay, so important things about viruses. We do not consider them cellular. They are not cellular. We don't consider them alive or dead. they're considered to be infectious particles. They can be active or inactive. They're incapable of replicating independently on their own. They are obligate intercellular parasites. They have to invade a host, sometimes a specific cell in a host, and use the genetic machinery inside your cell, use your ribosomes, your pool of amino acids, your transfer RNAs, use all of that to make new viruses. So viruses are made up to several things: genetic material, which is either DNA or RNA, but not both. So they're unique in that some of them can have RNAs, their genetic material. They can have either single-stranded or double-stranded RNA. Most RNA viruses are single-stranded. The DNA viruses can be double-stranded or single-stranded, but most of the DNA viruses are double-stranded. So they all have some sort of genetic material. The genetic material is surrounded by a protein capsid. The capsids made up these subunits called capsomeres, which can either be the same protein or a different series of proteins. And then some viruses have an envelope surrounding them. So this is the nucleocapsid, meaning just the protein and the genetic material. The envelope is a membrane that the virus does not make. They get it from the host cell that they infect. And then they modify the membrane by putting their own proteins in the outside of it. So viruses, they can be animal, plant, or bacterial. You mentioned before they can be DNA or RNA. Their shapes can be helical or icosahedral. The classification depends on their structure, chemical composition, their genetic makeup. So viruses families are viridae, and their genre, the suffix virus. So I won't go over these again, but these are just a wide range of different viruses. We're mainly interested in the ones that infect humans. So the genus Simplex virus belongs to this genus, and the human herpes 2 virus that infects Salmonella. There's the rabies virus. So those would be the species. Ebola virus is literally the species. So 24, go to 47. This just shows you after infection with a bacteriophage, the enormous number of virus particles that can be made. A single human T cell that's infected with the HIV virus, its burst number, which scientists estimate the number of virus particles it can release. A single T cell infected with HIV can release up to 50,000 virus particles. So as a review, the bacterial, so we already talked about this in the genetics of bacteria and DNA technology to a certain extent. So now we're talking about the virus, the bacteriophage virus. It either has a lytic phase or a lytic cycle. The life cycle of the bacteriophage in a lytic cycle ends in the destruction of the bacteria. In the lysogenic cycle, the bacteriophage DNA incorporates into the bacterial DNA where it's a provirus, and it will replicate right along with the bacterial DNA. To grow viruses, a lot of strains, animals that are used to cultivate viruses, white mice, rats, hamsters, guinea pigs, rabbits, and different sites in those animals can be used to cultivate specific viruses. We can also cultivate viruses in tissue. This is referred to as in vitro, in other words, outside of life. A technique that's referred to as in vivo would be inside of life. So tissue culture is a simple, effective way to grow populations of isolated animal cells in culture dishes. Most viruses are propagated through cell culture. Much of a virologist's work involves developing and maintaining these cultures. So we see when a virus infects a cell, these are the normal cells. They become abnormal. We call these cytopathic effects, and they can be characteristic of a virus. I didn't have this in the slide, but I'll just mention it briefly. Fresh primary cell cultures or freshly isolated animal tissues placed in a growth media. They undergo cell division, mitotic division, producing a monolayer on the surface, and they retain their characteristics from which they were derived from. Continuous cell cultures have altered chromosome numbers, so every time you propagate, in other words, subculture from one dish to another, you can have changes, can happen, alteration of chromosome numbers, they can grow more rapidly, changes in the morphology. And then I just had the last slide, I had 67. Rather unusual. I'm not, did I actually include this? Maybe it's on the test. Other non-cellular infectious agents or satellite viruses. These are dependent on other viruses, replications. For example, adenovirus, which can cause respiratory infection, has an associated virus. Originally thought it could only replicate in cells. Infected with the adenovirus, we now find infected cells with other viruses or have had their DNA disrupted through other means. So not a lot is known about these satellite viruses compared to what we know regularly about viruses. So I didn't cover that. So exit out of this. Does anybody have any questions about any of that or anything else concerning the test? The test will be next Tuesday. I will actually I need to put up a couple for the last couple of lectures. I need to put up some review quizzes, and I have these recordings now on a YouTube channel that you can view. As soon as I figure out how to link that up, I will put it in an announcement. So are there any questions at this point?
No, I don't.
Okay, anybody else have any questions, or I'm going to end this? And
No.
Hope everybody has a good holiday.
Thank you.
too,
Same to
Professor.
you.
Thank you.
Okay, everybody have a happy holiday. Enjoy the nice weather. We will,
And thank you for doing a review. Thank you for the review.
sure, no problem. We will have a lecture too, before I forget that, after the test next week. So I'll include that in the announcement in the Zoom invite. Okay?
Okay.
Okay.
So anybody else, I'm going to end this